Hey, welcome back. So this week we're going to be discussing the high-level content that I alluded to in the dihybrid cross video. So if you haven't watched that one yet, it's best to start there. I've titled it When the Ratio Comes Out Wrong. So when we looked at dihybrid crosses, we've thought about the whole 9:3:3:1 ratio, which is when you have two heterozygous individuals crossed together. The other classic one that you might get would be a heterozygous and a homozygous recessive, which is also called a test cross. So when you use a homozygous recessive, so you can figure out what is the genotype of that other individual, because it could maybe be homozygous dominant, which means they'd all come out the same. Or in the test cross with a heterozygous individual, you'd get a 1:1:1:1 ratio, 4 ones. And then what happens if you don't get those ratios and you get something similar? So we know from meiosis that there's something called crossing over. So crossing over gives us genetic variation. It's one of the biggest driving forces behind that variation. We have which are independent assortment of the chromosomes, the way that they line up along the equator, and you also have crossing over, which gives us new possible allele combinations from the parental genotypes. Now, there's three things to know really when we're starting to think about linked genes, and that is, what is a linked gene first of all? So here, this would be unlinked genes. They assort independently in metaphase 1 of meiosis 1, and you end up with these four possible genotypes. Now, if the genes are linked, so here's big A, big B, little b, see they're on different chromosomes. A and B are on different chromosomes, gene A and gene B. Whereas here they're found on the same chromosome. The place where you find them is called the locus or the loci, and this would be linked. They are found together. You can still get those four possible outcomes, but the only way that you get the big A and the little b when they are, like linked together, is through crossing over. So you can see here, these two here, these are the less likely outcomes because they've only come when crossing overs occurred. And I'll look at a little animation of that in a second. And the other thing to note would be about the distance. We call it mapping units, how far they are apart from each other. So you can see here, P is closer to Q and P is further away than R. So there's a higher chance of P and R being found in what we call a recombinant. So these two here would be recombinants. And there's a higher chance when they're further apart that a chiasma would form in here. So the chiasma, remember, this is where that point of crossing over, multiple chiasmas always form. But if we want to mix up P and R here, there's a higher chance that one could form here than it could between P and Q, simply because they're further apart. So like you said here, far apart, many recombinants, close together, few recombinants. So recombinant, just think recombination, a new combination of ones that are of genotypes that are not present in the mother or the father. So then we have crossing over for that. So here's a nice little diagram. So remember, this is in my interactive lesson that you can get on my site for you to go back through. And what I like about this is that I've made it so that all those key parts like the chiasma, the linked loci of those genes, they can all be found for a click of the button, and it will highlight and show you the distance being like the distance here between A and B on the same chromosome. Now, in itself, crossing over is not a hard concept. You might be asked, like, what are the recombinants? And obviously here we can say that these two are the recombinants. You might be given a cross between two individuals that are heterozygous. What's the possible recombinants? And there you would have to identify these two. You might also then be asked, and what's the harder question, would be think about whether or not, like an easy one first of all would be like, where did those come from? Like, where has the variation come from when it's crossing over? To skip back up, it's obviously come from crossing over in meiosis. If you've got ratios that are similar to this, where there's a small amount of these other ones, then you can know that that's come from genes being linked. So these could be some simple multiple choice questions in an IB exam. A harder one to get our heads around is about chi-squared. So that's the test for association. We've got this horrible formula. You have a formula booklet now, But what that means is that the IB are more likely to ask you application questions on this. In a later video, we look at how we can link Hardy-Weinberg and chi-square together. But right now, it's thinking about like how can we apply chi-squared? You're no longer going to get given a mark for being able to recount the formula and like saying it's a test for association, which was in the past a possibility. So we have observed minus expected squared, and the other symbol to know, this is chi-squared, x squared here, chi-squared, and this is the sum of, you add everything together. So you want to make yourself a little table. I'm going to show you what one of those looks like now, and we'll skip back up. Here's our observed, expected, so we can see we've broken down each part of the formula. Observed minus expected, and then observed minus expected squared divided by expected, which is the whole formula, and it's the sum of all of those. Now, the degrees of freedom, which is where we end up with a chi-square value here, which is 0.47 in this instance here, in this example. We've got our phenotypes, and then here we're looking at like thinking what is the significance of this value here. So what we do is we look at our degrees of freedom, and we'll have a table and this is just an excerpt from the table, and this is a critical value of 0.05. We always use that value unless stated otherwise, maybe 0.01 sometimes, but in science we accept that if something happens 1 in 20 times or 5% of the times or 0.05 of the time, then we say that that's by chance. So we use that as our critical value of significance. Degrees of freedom is 3. How do I know that? Well, there's 4 possible outcomes, and that means that we do the number of possible outcomes minus 1. So here's our total, 0.47. Now it says here, we look at our critical value, 7.815. What does that mean then? So first we have a hypothesis. Our null hypothesis is the difference is not significant. The data fit the expected ratios. The alternative hypothesis is the data will be significant and that the genes are linked. So here we're expecting a 9:3:3:1 ratio. That totals, if you add those numbers together, that totals 16. So if I took this 556, that's how I've got these values here, the expected values. It's 556 times like 9/16 times 3/16 times 1/16 to get these values here. Now, of course, in reality, you can't have 0.25 in an individual, but we've taken a large data set and we're saying, is what we observe significantly different than what we'd expect? If it isn't, well, then we've got our dihybrid cross. We've got our unlinked genes that they're inherited independent of each other. Now, what's nice in this little example here, if I scroll back up, is if we have, let's look at a recombination frequency. So for instance here, we get some data where we'll just slide this over. And you can see what happens as we change. Now here, we've got a threshold that you can see that when it passes, like we accept a certain amount, it's 112 versus 88, we accept a certain amount of difference between the values. Now, if the genes weren't linked, we'd expect a 1:1:1:1 ratio. We've been told up above that it is a cross between a heterozygous and a homozygous recessive individual. So we'd expect 1:1:1:1 ratio. However, we can expect a certain amount of variation just due to chance because biology is random, things don't happen how they're always supposed to. That's why we need large numbers. However, if we start sliding this over, what we can see happen is what happens to our chi-square value. It's now 64. Our critical value is 7.815. If it's larger than that, well, then what we're saying is that, well, actually, because you can see this number's got bigger, this recombination value's got lower. Therefore, what we're saying, the genes are actually linked, that they're not recombining very often, that we're not finding these two occurring very often. And the smaller that number is, the larger the chi-squared value. And they're probably, logically, the closer those genes are to each other on a chromosome. And that means that we reject the null hypothesis and accept the alternate hypothesis that the genes are linked. So if you want to know what the steps are, we've got this summary here. State them, calculate them, work it out each, and then at the end, we're going to compare to the critical value. And remember that we look at the critical value because it's the number of variations. How do we know a degrees of freedom? The number of variations minus 1. With this, it's always going to be with a dihybrid cross. It's always going to be 4 possible outcomes. So it's always going to be 3. You're not really getting tested on that. Now, if you want some practice, I've got a table here that's interactive. You can change the values. You can see what happens. It starts with a set of values. You can see what we would expect with a 9:3:1 ratio, so 2 heterozygous individuals, and you can see what would happen in a test cross as well with those same values there, and we can see what's going to happen. If you want, like, you can actually do some counting yourself. You can add them in. You can check your counts, what they should have actually been, and then you can also send your counts up here and see what would happen. So that's a really nice little interactive activity there to test out your knowledge of chi-squared. There's 2 full problems here for you to have a go at, and with a work solution underneath. Now, I can't stress enough that it's not necessarily going to come up in the next iteration of them, but like, they've just put it in the data booklet that you don't have to memorize the formula anymore. So it makes logical sense that this is going to be tested, maybe in conjunction with Hardy-Weinberg. So stay tuned for the next video. And to just summarize before we finish up, how does this all fit together? Now, we know from meiosis, one of the driving factors that creates variation is crossing over. That's occurring in metaphase 1. It produces recombinants, so new combinations of genes. What affects the recombination frequency is how close those are together. And we we get a gene map for how far apart things are. If we look at recombination frequency, we can actually use that to make the gene map and figure out, and that's what scientists would do with like fruit flies. Thomas Morgan's the famous guy that penned all of this, and he was looking at how often the different genes recombine, and he was able to figure out how far apart they were from each other. And chi-squared is our statistical test that's going to be able to tell us whether or not genes are linked or found on the same chromosome. As always, at the end, I've got a fill in the blanks and some IB style questions. So a classic one, 2 genes are described as linked. What does it mean? Their loci are on the same chromosomes, so they do not assort independently. Now, as a reminder, if you want help learning all this jargon, then check out cramly.study for a free space repetition flashcard app. It has daily games. There's a daily game that's similar to Wordle called Helix, and that changes every day. There's a global leaderboard, so you can see how you're doing against other learners in the world. So join thousands of other students on that today. Okay, thanks for listening. And in the next video, as I said, I'll look at Hardy-Weinberg and linked genes in conjunction with each other. Ciao.